Welcome everyone to this last webinar in this series. The first two webinars were fantastic. We covered some scope and vision for AlphaFold in the first one, and then a discussion about the impact of AlphaFold on teaching and training in life sciences. In this webinar, we are going to have use cases, and so we wanted to cover some wide use cases, wide range of use cases, and so I think we have a fantastic panel here going to present a lot of different ways AlphaFold has been used. Today's webinar will be chaired by Janet, so I'll hand over to her and just mention that all these webinars are recorded, and there is a link here on your screen where all those recordings are available. Over to you, Janet.
Thank you. So it's my pleasure to chair this, and I think we ought to get going as quickly as possible. This, as Samir has just mentioned, is about research and development and use cases. And our first speaker is Jan Kosinski, who is a colleague from EMBL based in Heidelberg. And really over to you, Jan, to tell us about how you can use AlphaFold in accurate structure prediction.
Yes, so yeah, indeed we are it seems everyone now uses AlphaFold, and judging from the participation, everyone is interested in AlphaFold, and we use AlphaFold as well. And it actually was quite a breakthrough in one of our recent projects. And I want to tell you about this today, about this project, which is from the field of integrative structural modeling, which means which involves integrating various experimental data. And I will use this example to show you how AlphaFold impacts the entire field. And the use case is the nuclear pore complex, the human nuclear pore complex. To remind you what's that, it's a complex that is embedded in the nuclear envelope, in the membrane around the nucleus. And it's a huge complex composed of 30 proteins called NAPs. Each of these NAPs in many copies, reaching around 1,000 copies in total. The complex is gigantic. In total, we're accounting for 120 megadalton and also having 120 nanometer in diameter. So this complex cannot be purified, cannot be crystallized, and so far has, actually as a whole, has not been amenable to higher resolution structure determination. What's important for this talk that in this complex, we can distinguish two different main parts. One is the scaffold, a more structured part that binds to the membrane, fuses the membranes, and creates a scaffold for more functional parts, which we call filaments and nuclear basket, and so-called FG nucleoporins, these disordered spaghetti-like proteins in the middle of the channel, which actually form the permeability barrier of the complex. And while the scaffold accounts for 76 megadalton of this 120, those flexible parts account for the remaining around 40 megadalton. And since the structure is so huge, so flexible, and embedded in the membrane, this structure has been for years characterized with integrative approaches. So first, over the last decades, many structural biologists solved individual structures of the complex of proteins, subcomplexes, or individual domains. And in 2016, together with, when I was actually a postdoc with Martin Beck at TMB Heidelberg, we make this breakthrough in understanding the structure by combining electron tomography. The Martin Beck lab obtained electron microscopy map at 23 angstrom resolution, which still seems too low, right? It cannot be used to see structure at atomic level, but was a breakthrough at the field at that moment. And together with these crystal structures and many homology models that we built, so just to remind, homology modeling used to be this useful technique with which you could predict structures based on other structures, and also using extra experimental information from a technique called cross-linking mass spectrometry, which I think will be described later by Yuri. We combine this data in some automated way to obtain this, a structure of the human nuclear pore complex. And that structure was a breakthrough in the field, but actually accounted only for 30% of the entire complex. And so it was just a part of the scaffold of the complex. And since then, Martin Beck's lab was trying to improve the resolution of the complex, and by recent years, they managed to push the resolution of the nuclear pore complex to around 12 angstrom in some regions, using so-called purified nuclear envelopes. So it's not a purified complex, but kind of nuclei imaged under the electron microscope. And also they managed to image the human nuclear pore complexes directly in cells, obtaining another map, a 3D map, electron microscopy map of this complex, which is similar, but has a more native diameter, a dilated diameter of the complex. And Agnieszka Obarska, the shared postdoc between our lab and Martin Beck's, has tried to use these maps to update the model of the human nuclear pore complex. So what she did, she took the maps and then she tried again to build homology models based on of those components. It's been super difficult. Many parts couldn't be modeled with homology modeling, and what she was getting in the end was a better model, but only slightly updated, with the total mass more or less being the same. And it was the situation as of spring 21, or just before the summer 21. And then, as you all know, the revolution came. And of course, as soon as the database was online and the program was released, as soon as the database was online, we just started downloading the models of the nucleoporins from the database, seeing that all of them are there, and they have very nice confidence scores. From the previous webinars, I guess you already know that these blue colors indicate confidence, the local confidence of the models. So we could get all those models. We could also compare them with some crystal structures that were not yet in the PDB that we obtained from another lab, and we could see that the models agree with crystal structures very well, even though those structures were not in the PDB. So no training bias. We could take the structures and one after another fit to the electron microscopy map that we had. Therefore, validating the structures like this big protein just fitting into the density here. We could see that those structures are much better than the homology models that we had before. So on the left, we had our attempt of modeling the protein before with the homology modeling, so not a perfect fit and some missing parts, huge regions. And on the right, we have a AlphaFold model, which fits the density much better and also fills these missing parts. So we are amazed with that, but then another amazement came from CollabFold, so the community effort, which modified AlphaFold to model complexes. And you also heard about this already on these webinars. And then we quickly downloaded CollabFold and installed this on our EMBL cluster and started modeling different complexes. So nuclear pore complex is too big to model with CollabFold, AlphaFold in one go. But what we could do is we could take some parts of interactions that we knew from previous models or biochemical experiments and keep modeling them. And then we actually just kept modeling, basically getting a gallery of multitude of structure of subcomplexes of the nuclear pore complex. Some of them we already knew and just refined, some were completely new. Again, we could compare with unpublished crystal structures, showing that this peptide interaction that we predicted is just matching the crystal structure, or this more regular complex is also predicted almost is almost identical to the crystal structure. And also, we could repeat what we did for the monomers. We could take the subcomplexes and start fitting them into the electron microscopy map. And what you also saw that actually they fit very well, even if they are big or sometimes have arms, kind of showing that even though the structures are modeled outside the context as fragments outside to the context of the complex, they still bring the correct conformation. And we could quickly see that we can map some proteins that we didn't have before into in this protein, which is very critical protein apparently for holding the pore together. or this dimer of two proteins, which no one could ever crystallize, despite many attempts, and no one could localize them in the complex. And now we found where this transmembrane protein comes in and how it interacts with this green protein, which everyone thought is in completely different place of the complex. So what Agnieszka did then, I mean, she basically, this is the slide from her favorite slide from her presentations. She basically trashed the entire year of work of homology modeling, refining models, fighting for every little helix to extend the model and the fitting. She trashed that work and repeat the modeling now with AlphaFold pieces. So what she did, she took the electron microscopy map, this high resolution map from Martin Beck, and now all the monomers and subcomplexes as new pieces. And then she used a special software that we developed in our lab, which is called Assembline, which is an assembly line of macromolecular complexes. And this program can take those individual pieces and find optimal configurations in the electron microscopy map. Plus, we added some new components to the software, some elastic networks that can use AlphaFold models and the interfaces from AlphaFold models more wisely. And then, since we had this insert structure, but that was at lower resolution, Agnieszka took this constricted model and then refined using Assembline into this dilated structure. And Yuri, forgive me for this comment. So what I want to emphasize here, that actually, so far we always had to integrate data to build these models, but in this case we actually didn't use crosslinks anymore. I don't want to say that crosslinks are not useful here. What I want to say that the idea of integration is changing a bit. It might be that now we can integrate less. We might be able to go ahead with just single data set. Could be that even our electron microscopy map is not necessary, but with some crosslinks and AlphaFold models, we can actually get good predictions. So just summing up, so this is what we got. So this was the model from 2016 that covered 35 megadalton of 120, and given that but the total mass of the human nuclear pop complex is 120 megadalton, and the new structure covers 70 megadalton of the complex, which is around 90% of the scaffold that I showed you at the beginning. And this work has been just published a couple of weeks ago, so you can read exactly in detail how we've done it and how we also validated AlphaFold models, how we checked all the interfaces and so on. So what's next? So this is again the outline of the nuclear pore complex, and what we've been doing over the recent years together with Martin Beck is a stepwise approach to build the model of this complex. We're more or less done with the step 1 and 2 for the scaffold, but still there are remaining parts, these filaments and the nuclear basket. And it seems that AlphaFold actually can help with those structures, and we have some interesting leads there. And the last step is these disordered regions here, but it's probably is at the moment outside what AlphaFold can do, and this is a subject of other modeling and experimental approaches. So the modeling was done by Agnieszka Barska, and molecular dynamics for the work, which I didn't show today, was done by Mark Ziegel, and this is a long-standing collaboration with Martin Beck, formerly from EMBL Heidelberg and now from Max Planck of Biophysics in Frankfurt.
Thank you very much indeed, Jan. That was a very clear and comprehensive description of how you can integrate the AlphaFold models into this sort of modeling of these giant complexes. So there's one specific question in the Q&A, and it said, I'll quote from it: "You mentioned homology modeling, produced models are not so good, not having a ground truth. How do you know whether it's good or not?" And yeah. I might add to that. The approach seems to be to divide and conquer, i.e. you do bits, you do pairs, and then you put it all together. How many places do you get changes by integrating those pairs into the whole thing? And that relates to the quality. Do you see what I'm trying to ask?
So for the main question, so how do you know it's a ground truth, and how do we judge? So first we judge from of course, from scores provided by AlphaFold.
Yeah.
So AlphaFold provides a variety of scores that allow to evaluate the local quality and also the quality of interdomain orientations. This is the first thing. The second thing is that because we have a lot of experimental data, so we check with the electron microscopy map, and that even if we don't know where it fits, we actually find a place that agrees with the model very well. We have some statistical procedure to check whether this can arise by chance or not. And the third thing, which I haven't shown, but is I think half of the supplementary data, we actually analyze the interfaces according to some physical properties and other scores, also machine learning scores that assess the likelihood. And we compare it to some known negatives, so some artificial pairs that we can distinguish that. But I didn't get your question, Janet.
Okay, so you've got a pair of proteins. You model that pair and find an interface, but that's just for the pair. It's not in the context of the whole pore.
Mm-hmm.
So to what extent do those pairs get modified by being in the whole?
So what's amazing that often those pairs just fit like a knife in the butter. Yeah. So the AlphaFold seems often to model conformations in a bound form. I always hypothesize that this because this is the functional form which is encoded in the alignment and which is constrained, and that's why we get it. It's not always the case, especially that in the nuclear pore complex. Sometimes you have different copies and they have different environments, and sometimes they do need to change. And then we have some approaches to cut them into smaller pieces and optimize a bit better. Okay. But overall, they fit much better than homology models based on crystal structures or cryo-EMs or isolated cryo-EM.
Yeah. Okay, so there are some other questions now, but I think we're at the end of the 15 minutes. So if you could answer those questions, Jan, in the chat.
Sure.
Mm-hmm. Our next speaker, then if you take your screen down, and if Juan Carlos is our next speaker. So Juan Carlos Mubarak, he comes from AstraZeneca based in Cambridge, UK. And he's going to most importantly tell us about how AlphaFold is being applied in the pharmaceutical industry. So over to you, Juan Carlos.
Many thank you, Janet. So let's start saying that in pharma, we've been using models for different applications for at least 20 years. So now we're very happy to have the new AI-based methods like AlphaFold to predict protein structures. And let me start saying that those AI-based models could be applied to any application where experimental structures are usually applied. So it could be to just have an idea, for example, how a molecule is working, what is the mode of action. It could be, for example, also for lead optimization as well. It could be used for virtual screening. There might be an endless number of ways we can apply those models. And of course, the more accurate those models are, the more applications we can use them. And when we got, AlphaFold appear, I mean, the first things we wanted to check is like an internal control, right? To see whether AlphaFold were able to perform well as well in an internal challenge. So we look in our database, right? A structure which will not be publicly available in the protein data bank. And we're 100% sure this structure was not there. And AlphaFold nicely predicts the structure to a high level of similarity to the one we have in the internal structure. So, and on top of this, there were regions with low electron density. And of course, AlphaFold predicted a possible loop conformation, which was quite reasonable. And I have to say, other programs can also do the same. But also, as well, for a region where on the crystal structure was ambiguous secondary structure, AlphaFold was able to predict a defined beta sheet structure as well. So in our opinion, this is unprecedented accuracy for a structural prediction task. So we were quite happy with this. And I want to show you to you as well another example of lead optimization. So in a case where we're trying out to test AlphaFold multimer, no? The function of AlphaFold to predict the protein, not just the model, but also the interactions among them. So make a complex prediction. So in this case, we had a peptide, right, that which was selected from a library, and we also knew the target. No? So, and of course, we wanted to see whether AlphaFold was able to predict the structure of the peptide bound to its target. So first we used the AlphaFold 2, the first one, we call AlphaFold monomer here. And if we want to predict a complex, the receptor is in green and the peptide bound was in cyan. We then later on tried, I have to say that in this case, we include the technique of using a glycine linker. I'm not showing here the linker, but we connected the receptor to the peptide with a polyglycine linker because the first version of AlphaFold was also able to predict multimeric structures. And this is the result we got in that case. Then we used the new AlphaFold multimer. In this case, we got a prediction of the complex bound together. And then we also tried to use industry commercial software to make the same task. In this case, we predict the protein, the target, and the ligand, and we dock them. And also we use a combination of different programs, as we often do. From one method, we predicted the structures, and with other tools, we predicted the docking. And we got those three highly similar, let's say, predictions, although I have to say, because AlphaFold, in some cases, we got this, let's say, orientation upside down. So we look, let's say, as the same you see here, but the other way around. And also with one of the computational tools. We'll try different other methods as well. I'm not showing this figure as well, but those other methods are also were able to predict those alternative binding poses. Now, we went on and actually we predict experimentally the crystal structure of this complex as well. So these are now our new internal structure. We did this after we built the models. And you can see here that the predicted crystal structure looks quite similar, let's say, to different cases. You can see here, just measuring only for the receptor RMSD, and for multimer got the lowest RMSD, lowest deviation with the ground truth. Now, if we compare for the ligand RMSD, right, the cyan one, so you will see here that AlphaFold did a quite good prediction. We got a slightly better one with a combination of commercial software as well that we normally use in industry. So I have to say that in this case, AlphaFold produces the most accurate receptor, but commercial software could do equally good in this task, right? Of course, there are other more complex tasks where might not be the same case. So I think we are quite impressed by the performance of the new methodology as well. And we have been trying to apply this. It was a case of legal optimization, where now that we have the structure, we can use those models and structures to start optimizing those ligands. We went on to more complex challenges as well. We try now to predict with AlphaFold multimer, right, the interaction between protein-protein interactions. And we have a case where we're trying to predict whether AlphaFold was able to tell where there's the right binding site, right? In a case where there was no crystal structure available, and we predicted the multimeric complex using AlphaFold 2. So the model was also, let's say, after we got the model, we also ran long molecular dynamics simulations as well to see whether the predicted model will stay in place for around 2 microseconds. And also we got, I would say it was a challenging target because attempts to get cryo-EM structures didn't work out. We only were able to get two-dimensional, let's say, cryo-EM electron microscopy, two-dimensional average, how they call, no, they're like pictures, of the target on the grid, and that's the only thing we were able to compare with the AlphaFold results. And you have here in this figure, we have this electron density, right, after the simulated model. And you can see the receptor is on the bottom, and the target protein, let's say the protein that is the ligand, right, is bound in a one particular place and was stayed there during long molecular dynamic simulations. And we compared this with the cryo-EM electron micrograph, and we're able to tell by the 2D pictures that the model looks fairly right. So we also are quite impressed for this case where we can understand better the mode of action using AlphaFold and getting a 3-dimensional version of a 2-dimensional experimental data, which not always is able to predict the 2-dimensional structure. So So we're doing in time, we're fine. So I have to say that, as I said before, the AI models can be used for any case where experimental structural usually applied. However, a structure-based design for the purpose of drugs, I mean, it's very sensitive to details. So I'm just playing here a hypothetical example, no, where if we have a model where a single key amino acid side chain is in a different or wrong conformation than, for example, would have been predicted by an experimental method. I mean, this could make a, let's say, a domino effect, no, on which then the further applications on the pipeline, right, that are usually being designed to work with perfect crystal structures will have a deep effect. For example, you can imagine a virtual screening. A wrong residue in the binding pocket will affect a lot the kind of molecules being selected. So, let's say if we were to compare, for example, experimental structure with the AlphaFold models and other models, I would say generally the accuracy of AlphaFold will be slowly lower or unpaired with experimental structure. Another kind of model will be to a lower degree. However, the potential error, right, that might go, the propagation, of course, will be the other way around. No, we'll expect to have less propagation using a crystal structure than in the case of a high quality model, and the lower the quality of the model, the more chance we have to have, let's say, compounding errors later on the pipeline. So the take-home message is that all those AI-based workflows like AlphaFold and others, I mean, are already being applied to early R&D at different countless cases. I mean, they could be equally good or better at predicting structural models than programs we often use in industry. The accuracy of the models greatly dictates the applicability of those that can go from idea generation up to molecular design. And a small inaccuracy, let's say, in models that apparently could be tiny, for example, like only a rhodomer in the wrong conformation, could have a compounding effect in further applications for structure-based drug design. So, and I think we should keep improving those computational methods because they work fantastic. So, acknowledgement, people on the company. Thank you.
Thank you very much. So that's really proof, I think, that AlphaFold can be helpful, but we should include other technologies that have been developed before, specifically for the small molecules. So, question for you. Do you think that industry will turn to AI models instead of experimental structure determination?
I think we're using both. We are using experimental determination, and on top, and as I'm sure as well, we're combining experimental determination like cryo-EM as well with AlphaFold. So I think the fusion of physics-based techniques with AI methods, I mean, can be used all together, right? And as well, if there's an urgency to start quick with a project, you can always start first with a model while the structure's going to be solved at some point. So
yeah. So if I could ask you a question from me, you showed us examples of protein-protein and protein-peptide. Have you tried doing anything with the small molecule ligands that aren't proteins and peptides? Have you, to any extent, thought about that? How do you think these methods might develop in that direction?
So let's say the tools that we use in industry for structure-based design are mostly made, as I say, for crystal structures. So many of them assume the receptor as rigid, right? Yeah. There are a few tools, of course, that take into account flexibility, but not the most. So, for example, for virtual screening, right, or structure-based design, it will assume the protein as rigid, right? So any, as I say, any tiny accuracy could be on the binding side, right? It will affect which kind of molecules are being selected by the shape fitting mechanism. So I have the impression that even though the general fold might be correct, if we try to go to the details, I mean, we will need a higher level of accuracy that the current level the method are doing for doing further on drug design, structure-based applications.
Yeah. So a question here. So are structures and structural studies useful only internally within the company, in the design process, or expression regulatory agencies asking for structural proof that your new compound is working as expected? I don't know if you could get that. So basically, can you know, how does it?
Yeah, so I guess the question is related to whether somehow the structures can help to prove the mode of action. If I understand the question correctly,
Yeah.
Yes, often the structures are used as a proof of mode of action. Now we have the structure, we have the ligand bound to it, and we can say this is the way it works, right? Now, if the model is as good, we have high confidence that the model is right. They have the potential to be as well used in the same case, but it won't be experimental proof though. So we have to be careful there.
Yes, we have to be careful, I think. I think an easy question to end with, can we do molecular docking with drugs onto targets modeled with AlphaFold as well?
Yeah, we can always do them. I mean, the question is the accuracy, right? How accurate are those and which result we're going to get? I mean, so as I say, we've been doing that kind of screening for decades in industry, right? We have plenty of software available. And as I say, they work best with crystal structures, and then the models go down. If we have a model that is as good as a structure, we expect to get as good results. I mean.
Thank you. Thank you. So I think we'll close there. That was very, very nice.
Thank you.
So our next speaker is representing the cross-linking mass spectrometry community, and Juri Rapsilber, who comes from the Technical University of Berlin, is going to tell us about his work. So Juri, over to you.
Thank you very much. Very exciting seminar series, and I'm honored and awed to be asked to present our work here. And let me start by picking you up on what others have said already multiple times, and what Catherine pointed out very early on 2 weeks, that AlphaFold is great in giving really high quality models and in giving a very good estimate of how reliable they are. So it's two things, giving good models and giving good confidence or accuracy of confidence. Sergey showed this protein model here from YETE, from Bacillus subtilis, and said, well, the similarity of the structure of this uncharacterized proteins with a known protein allows speculating on what this protein might be doing. And then it was Randy who in fact pointed out that well, you can take that to the next level. You can just like predict all the pairways and whatnot interactions across the genome, which then would of course also give you some insights on what these uncharacterized proteins are doing. Now, if we do this just for fun for Bacillus subtilis, which has like 4,100 something predicted protein coding genes, then you end up with 9 million pairs. I don't know about your computational resources, but if you then remind yourself that it's not only 2 proteins that may interact, but also 3, then you end up at 37 billion and so on and so forth. So doing this exhaustively without any other assumptions or input is probably going to be computationally challenging. And because that would give you only the models, the question then is also, what about the accuracy? So it's always model and accuracy. It's the pair of the two. Of course, we don't have to pretend we are dumber than we are, because there is information about protein interactions, and we could use that. Systems biology, including proteomics, gives us information on proteins that are functionally interacting, for example, being in the same pathways, or proteins that are physically somewhat interacting by being in the same complexes. Now, structural biology is much more tight than that, and that includes AlphaFold. It tells us 2 proteins physically are with multiple residues within like 5 angstrom. And interestingly, there's a technology that gives you of this order of resolution, and that is cross-linking mass spectrometry. And of course, I disagree with Jan on the usefulness of the technology, but we can discuss this later. Now, being at a comparable spatial resolution level to structural biology, cross-linking mass spectrometry data places itself in a pole position to kind of like provide candidates for AlphaFold prediction, and also maybe to validate those predictions. Because after all, I mean, they are models, and it would be good to give at least some experimental evidence as to their existence. All right, good. Before we go to our data, let me just quickly pick you up on what the technology is about. When we do cross-linking, we add chemicals that are bifunctional, so they are reactive on either end, and they do react with some specific side chains and proteins. And then we take a proteomic approach. We digest all these proteins, have some peptides that are linked and some that aren't, acquire mass spectrometric data on these, interpret the mass spec data to then get ultimately this pairwise information of which residue from one protein is close enough to which residue of another protein or itself, in the same protein, to be linkable. And this then can inform modeling and further experiments. One nice aspect about cross-linking is it's not limited to isolated proteins or complexes, but you can actually do this also with entire cells. And while, of course, then the analysis is a bit more taking, what you do get back is then a network. And don't forget that in this network, where every dot is kind of like a protein and the links tells these proteins were cross-linkable, you still have this residue level resolution. So we do know which residue of this protein was close to which other residue of this other protein. So that suggests that we could do kind of like a proteome-wide screen, or attempt to do that, in order to then feed in these interactions into AlphaFold and then come back and use the crosslinks to validate our AlphaFold models. But I told you it's not only about the models, it's also about how accurate the models are. And the same is true about the data. Now, we have the data, but how accurate is this data actually? And as it so happens, the cross-linking field has been working towards developing error estimation procedures as well. However, it has not yet arrived at one coherent view on this. And let me show you here a data analysis we did for E. coli lysate being cross-linked and analyzed the way that I presented to you. And to test these different approaches, what we did is we interpreted the data not just against an E. coli database, but we combined a human database of same size and same size distribution of the proteins with this E. coli database. And if our analysis is good, we should not find any human proteins, of course, because they are not in the sample, they should not be in the data, and henceforth they should not be in our interpretation of the data. And if we do take one accepted way of doing error estimation by cross-linking, this is how it looks like. In fact, we do see quite a lot of human proteins in there. And mind you, this is the known random ones. Of course, among the E. coli proteins, we have an equal number of wrong E. coli proteins that we actually don't have real evidence on. It's just down to not quite correct data processing and data analysis. However, the good news is bad news. The good news is, well, there's procedures to do it better. So we can do it in a different way. And if you're interested how that exactly happened, it's the paper reference that's given there at the bottom that I would recommend you to read. You do get a smaller network here in this case. Let me just rearrange that. It's not free of error. That's not what I promised to you, but it is a controlled error. It's a known error. And we can tell you, in this case, we were happy with 5% error. And we can count the number of proteins in there and tell me if it's telling up to 5. Of course, when you have only one, that is a bit of an extrapolation, but it is very reliable data that we do get back. So we do get interaction data, and it is very reliable. And this we then threw on, incidentally, Bacillus subtilis, and that's our first network data that we did get back. And the colored proteins, or the bluish purple colored proteins, that are in fact proteins of unknown function that we would like to understand what their function is by linking them through cross-linking in cells and then through AlphaFold models to proteins of known function and to understand what their function is. And don't forget that crosslinks always are binary information, so we don't know the higher order makeup of the complex, and therefore we have combined this data set with some fractionation data of complexes where we separated them by size exclusion chromatography, which sorts proteins by size chromatographically and then identified in different fractions. And we did this for untreated cells, and we did this for cross-linked cells. And you can see here in one example that a known interactor NAS-A of the RNA polymerase is in fact through cross-linking stabilized and then is detectable as an interactor or co-migrating. And the same is true for an uncharacterized protein, so that are missed without covalent stabilization of this complex in the cell before doing this. Okay, so we have this data, and of course we also have literature knowledge. And the literature knowledge we used was in form of ZubTiWiki, which is a geniusly maintained resource by Jörg Stülke, who's incidentally also our collaborator in this entire endeavor. So we have three sources of information that we put together. We had our crosslink data, we had the coelution data, so it's some 240 crosslink pairs, some 670 coeluting pairs, and then some 1,000 protein pairs that we took from ZubTiWiki. We predicted those using the AlphaFold pipeline, and you can see IPTM over PTM scores that we get kind of like two clouds, and that's already very suggestive, kind of like this is maybe the failures. No, that's the random cloud, and this might be the signal, but we don't know where to cut. No, so that's our problem. Now, what is now the data that is reliable here? What other models are reliable here? And we approached that by throwing also some pairs in there, like we did with our, remember, with the human E. coli mixed database. We did the same here. We took bacillus proteins and paired them with random E. coli proteins. I mean, we know they are not in a complex. So what does AlphaFold make out of this? And this is the IPTM distribution that we get. In red, it's our negative control. And in this purple color, that's our, well, it's not positive control, it's our data, our Bacillus subtilis pairs. And we can see that at the high IPTM scores, none of the random or negative control complexes are scored. But even at fairly, I don't know, at IPTM ranges where probably people would say in the moment out of gut feeling, don't trust this, there's still a surplus. So there seems still to be useful information there. Anyway, what we from here on forward in the presentation look at is just this bit here. And we do have crosslinks. So we can see that at the high scoring range, the crosslinks do fit to the models, whereas at the low scoring range, the crosslinks disagree. So this is violated crosslinks here, and the mediate models where it's the mixture. We have that, of course, per complex, and you can see that, so this is color coded here. Dark means less agreement of the crosslinks with the models, and you can see that the light colors are floating up here at the high quality model. So crosslinking does support what AlphaFold gives us here in dimeric complexes. Focusing just at the absolute top here and only at the novel interactions, we get like 17 novel interactions that previously weren't even known as interactions, and we have them here now as structure models. Let's look at a few of them. It's now soluble complexes where we do have supporting evidence then for crosslinks that wasn't used in the modeling phase. So we can still use them here to validate the models that we got back. And we also see that for membrane-bound complexes, and nicely also for cases where we have heteromeric complexes of paralogous proteins, which are maybe a bit more challenging to predict because they could also be homologues. Good. And we not only had, of course, pairwise interactions in our network, we also have multi-protein relationships here. And if we look at one example, the pyruvate dehydrogenase complex, which is cross-linking here to an uncharacterized protein, then we see that this YNER uncharacterized protein gives a nice model according to AlphaFold with PDHA, and it also cross-linked to PDHB. However, that is, well, it's a poor model. We do know, however, from the coelution experiment that three proteins are coeluting. So they are forming a complex together. And in fact, if you put them all three into the multimer prediction, then we do get a fairly confident prediction according to AlphaFold that looks like this. It fits to our crosslinks. And in fact, this protein occludes access to the active site here, which suggests it might act as an inhibitor. In fact, if you overexpress it in cells, in bacillus cells, and grow them on pyruvate as an energy source, then they don't grow so well with the protein overexpressed. And we did do the control. It's not the overexpression of the protein per se, because in glucose, it's all fine. And with this, I come to the end. Was two PhD students who did a lot of the work, Rika from Jörg's lab and Christian. And Andrea did all the modeling work. And Francis, who also was an essential component of the team, is now in the US. So if you're into such work and that excites you, contact him at the NIH. I'm sure he could find a spot for you.
Oh, thank you very much, Yuri. So there are several questions for you. Perhaps I'll start some of them from Mohit Misra. What is the best cross-linking agent to start mass spec cross-linking? So where do you start? Which is the best agent? Right.
So there's hundreds of reagents, of course. And that can be a bit difficult to get the head around. I would say it depends still, it depends a bit. So if you have a purified protein complex and let's say you have an EM density and you want to interpret this EM density and fit proteins into it, and what you would like to have is good contrast. And I explain to you in a second what contrast means here. You want to have high confidence in that you see the canonical sites, not due to flexibility. And there, in fact, photo cross-linking gives you the best reliability and the best data by far. If you work on more complex mixtures, then activated acids like DSSO or BS3 are also, are the better ones in complex mixtures. If you do look at the results of the two, why are they different as such? So with the activated acids, they are not so hydrolyzable. They don't hydrolyze that fast. Of course, they do hydrolyze in water, but not that fast. They wait for protein to pass by. And that means you do get links everywhere where it's possible. So, and that becomes more difficult to interpret. And that is the lack of contrast. So anything that can come close to each other, you will find crosslink. And that will then give you a less easy time when you need to interpret the data in the context of one structural model rather than something that's very floppy and flexible. And there, the photo cross-linking gives you a much cleaner image because that reacts with water if water is around. Yeah. It doesn't wait for protein to pass by.
So a second question from Anya Rojas. Could cross-linking and mass spec be useful to detect novel amino acid type of linker interactions like lysine cysteine?
I'm afraid I did not understand the question fully.
So Anya, I think, is asking whether cross-linking approaches, and in particular mass spec, could be used to detect when you have novel linkers naturally between two types of amino acids.
Okay. So like lysine and aspartic acids as known linkage, but could one discover new linkages? Yes, of course. What is a big challenge is always this signal to noise, because if you're looking at the whole cell level, you have a lot of not cross-linked peptides, and then you want to look at a few cross-linked peptides. So somehow you have to beat that signal to noise.
Yeah. Okay.
I think that's the biggest challenge. There's an enrichment-wise, there is not the magic bullet yet to isolate these bridged peptides.
Okay. I'm suddenly conscious of the time, and I think we're at the end of your time, so I should there are some other questions you might like to answer in the chat. So thank you very much. And we move on to our last presenter, Katja Luck, who comes from the Institute of Molecular Biology in Mainz. And Katja is going to tell us a little bit about folded regions and disordered regions, I think, some applications that she's been working on. So Katja, over to you.
Thanks, Janet, for the introduction. Let me just move some windows around. I hope you can see my screen okay and my pointer.
Fine.
Okay, good. Then hello, everyone. Yeah, it's been for about 2 years now that I started my own mixed dry and wet lab at the Institute of Molecular Biology here in Mainz. And this is really to pursue my long-term research interests, to study proteins, how they bind to each other, and the molecular interaction networks that they form. And really, in this sense, the release of AlphaFold about a year ago really had a tremendous impact on my research. And in the next 10 minutes, I would like to highlight 3 achievements of AlphaFold that are impacting and will certainly impact even more in the future how we're doing molecular biology research. Oh yeah, there is. And so I start with my first achievement, or with the first achievement of AlphaFold. Now, what's in your mind actually when you hear the word protein? I think that in the head of many scientists, when they hear the word protein, they'll think of something like this, like a beautiful tertiary structure of a folded protein. However, it's by now really well established that proteins not only consist of these folded parts, but also of regions that are intrinsically disordered. And now these regions of disorder can be well predicted by tools like IUPRET, as you can see here, but it can also be identified in multiple sequence alignments, for example, where these disordered regions tend to be less conserved, and they differ in the amino acid composition compared to the more structural parts of this protein. For example, the structure I'm showing you here on top of my slide corresponds to the C-terminal folded part of this protein here. Now, the disordered regions in proteins really carry important cellular functions. For example, they can bind to other proteins or DNA or RNA molecules. They carry sites for post-translational modifications, and they can drive liquid-liquid phase separation. Actually, it's estimated that about 25% of the human protein is predicted to be disordered. However, these disordered regions, they don't crystallize. And so we really lack sort of a structural and visual representation of these disordered regions, why I think that in the heads of many scientists, still the sort of tertiary structure biased view on proteins persists. And so in my opinion, therefore, like one of the first and probably most unintended achievements of AlphaFold is actually the visualization of not only the folded parts of proteins, but also of the disordered regions in proteins. And so in the long term, I really hope that these structural models of these full-length proteins will really change the picture that we have in our head when we think about proteins. However, there's one caveat to this, because ironically, like any static structural representation of a disordered region of a protein, must be wrong, right? Because by definition, these disordered regions do not adopt a particular fold. And so while these structural models are really helpful to visualize these disordered regions, we should not make any assumptions based on the particular conformation at which these disordered regions are displayed in these structural models. Okay. And with this, I'll come to the second achievement of AlphaFold. If molecular biologists are trying to study the function of a protein X, then what they often do is they want to identify putative interaction partners of protein X using mass spectrometry, for example. They then pick an interesting interaction partner, protein Y, and they'll confirm the association between protein X and Y using, for example, a targeted pull-down experiment. They then would like to know what are the regions in X and Y that are mediating interaction. And to study this, they are designing protein fragments, which are then assayed again for interaction with each other of protein X. Now, a continuous challenge in this workflow was the design of protein fragments that are still properly folded, that are well behaved and well expressed. And so what scientists or researchers really need to achieve this is they need to know the exact boundaries between the structured and the disordered regions in their proteins of interest. Now, of course, the sort of predictors like IOPRED were quite helpful with this, but it was difficult to know, for example, for short regions of disorder, that they were actually like separating two different folds, or whether they represented, just a longer loop within one larger structured region of the protein. And also, it's kind of hard to really guess what the defined, like precise boundaries or start and end positions are of a folded domain. Now, tools like SMART or PFM allow us to predict the occurrences of known, but not of unknown types of domains. And oftentimes, the certain end position of the hidden Markov model matches do not correspond really well to the actual certain end position of the folded regions in the protein. And so AlphaFold, in combination with existing protein sequence analysis software, I think is now really a powerful tool to predict the precise boundaries of folded and disordered regions in a protein. And luckily now, these structural models of these full-length proteins are now easily accessible via the AlphaFold database, or by running your own predictions using the AlphaFold CoLab implementation. Now, this brings me already to my third achievement of AlphaFold I want to highlight today, but I've put a question mark here, and that's because I think that there's still a little bit of development that is needed here to for this application of AlphaFold to really take off. Now, AlphaFold was really designed, first of all, to predict the structure of individual proteins. However, the biophysical forces that drive protein folding and binding are actually quite similar. And so researchers really quickly asked the question whether AlphaFold can actually also predict the structure of two interacting proteins. And indeed, numerous studies quickly appeared where researchers benchmarked AlphaFold and applied AlphaFold for the prediction of structures of interacting proteins. And actually, DeepMind even published another version of AlphaFold called AlphaFold Multimer, where they adapted AlphaFold specifically for this application. Now, there are two common modes of binding, let's say, by which proteins can bind to each other. The first one consists, or is like a domain-domain interface, where two folded domains of one of each interaction partner would bind to each other. The second mode of binding consists of folded domains that bind short linear motifs, which occur in disordered regions of proteins. And now, using curated sets of structurally solved domain-domain and domain-motif interfaces and randomizations of these data sets, we, in line with published findings, observed that AlphaFold can actually predict reasonably well both modes of binding. Now, encouraged by these prediction accuracies, researchers actually applied AlphaFold onto human protein interactome data sets to try to generate structural models for like thousands of protein interactions. Unfortunately, though, the studies that were released so far only resulted in confident structural models for a small fraction of the tested interactions. Now, there's certainly many reasons for this apparent discrepancy, but one of the reasons that we identified was that benchmarking is often done on sort of minimal interacting fragments as they're also crystallized, while full-length proteins were actually submitted for the structural annotation of the human protein interactome. And so we observed actually, like, we can't really quantify it at the moment, but we repeatedly observed that AlphaFold is really successful when we are submitting for interaction prediction or the structures of these. We're submitting smaller interacting fragments. AlphaFold often fails if you're submitting pairs of larger protein fragments of full-length proteins. And so to overcome this challenge, we developed a pipeline where we're submitting fragments of interacting proteins to AlphaFold. And we are then sort of scoring the recurrence of residue pairs that we observe to be in contact with each other in generated structural models. And now, so we're in the process of sort of improving this pipeline and adapting it to be applicable at larger scale. But today I can present to you a promising example that we're working on together with another group at our institute. So our collaborators, approached us with a pair of proteins that I'll call here protein A and B that they found to interact with each other. They also had some experimental data that suggested that the second of the two red domains would actually bind somewhere in the C-terminal disordered region of protein B. Now, in line with IUPRET predictions, AlphaFold actually predicted the third occurrence of this red domain, and it also found that actually this type of domain here would rather be there in a particularly extended fold. Applying our fragment approach of submitting these protein fragments to AlphaFold, we actually repeatedly observed specific binding of a linear motif at the C-terminus of protein B to a particular part of the second domain of these three red domains here. So while we are awaiting experimental validation of this prediction, this would represent a novel mode of binding to this type of domain here. This, I'd like to thank my lab members, especially Xiaopian Li, who's doing most of the work with AlphaFold in my lab. Also want to thank Balint and Ora for insightful discussions on using AlphaFold, and the IT department at my institute for implementing a local version of AlphaFold. Of course, I'm very grateful to DeepMind for having developed AlphaFold and making it freely accessible, and to the EBI for setting up this AlphaFold database together with DeepMind. I also want to thank Martin Steinegger and his collaborators for putting AlphaFold on Google Colab. Really want to thank the organizers for inviting me to contribute to this webinar, and everyone for your attention.
Thank you very much indeed, Katja. So we have some questions, but I'm not sure how relevant they are. There's one about membrane proteins, and it says, in the case of membrane proteins that are embedded in a hydrophobic membrane, does the dynamic nature of the protein, sorry, it's disappeared now for some reason from my thing, does the dynamic nature of the protein affect your predictions in a way? So if you think about these short linear motifs, and sometimes they often do bind to membrane-bound proteins, is that environment going to have an impact on what you're trying to do?
Yeah, so we haven't tried that specifically, but what we have observed when looking at the predicted structure of a membrane protein, that AlphaFold wouldn't get it right, that there's an extracellular and an intracellular part that you wouldn't be able to touch each other. It would kind of make a kind of more globular kind of thing where these parts would be, touching each other. So in that sense, the structural models of membrane proteins might globally be wrong, while of course individual folded parts of the extracellular intracellular part would still be right. And here I think it would be even more important to apply rather a fragment approach to see which parts of the two proteins would actually bind to each other, and that should also then work for linear motifs.
Okay, so there's another question appeared. and as the questioner Marina Calandret says, the disordered regions are often low IDDT scores, so they're very low reliability. They're also often not physically realistic, so they've just been joined together two ends, so they don't really represent anything like a loop would adopt that fold structure. So do you think, given that, how can the AlphaFold, I can understand when in the interaction, when you get two proteins interacting together and a very disordered structure becomes an ordered structure. But actually, how do you think of these disordered regions in the context of what you're trying to do? I mean, are you thinking that they adopt a structure and that AlphaFold can help with that in the complex?
Yeah, so I really focus on particular parts within disordered regions, which are these linear motifs. So these are the binding sites that disordered regions have to interact with other proteins, folded domains. Yeah. And it's really interesting and in line with what a previous speaker said. So oftentimes when you look at these disordered regions in a full-length structural model from AlphaFold, sometimes there's suddenly a little helix appearing. Yeah. And sometimes AlphaFold actually detects that the linear motifs usually adopt a secondary structure up on binding to their partner, but they would not have this fold or the second structure when they're in the unbound state. But AlphaFold tends to predict conformations in their bound state, and you sometimes see like a helix suddenly in a disordered region of an AlphaFold model, which probably is the bound state, which is very interesting. So obviously, I think the conformations we get from AlphaFold about disordered regions are wrong in the unbound state, but it can hint us also to where linear motifs are, which would adopt this conformation up on binding.
Yeah, very good. Thank you. There's some more questions, but I think we should open up now for the general discussion. So if everybody could come online, yeah. I'd like to start off. We're lucky that Randy Reed has joined for the panel. So I'd like him just to make one or two statements, perhaps only one, Randy, about the how you see research and development coming out of the AlphaFold.
Yeah.
Where will it have the biggest impact?
Yeah, I probably said most of this in the talk that I gave in the first webinar.
Yeah.
And I'm just picking up on things that most of the speakers here have talked about. But I think what Katja was talking about, using AlphaFold to decide on your construct boundaries, that's a really big thing that every experimental structural biologist I talk to is doing. So that's important. In terms of structural biology, I don't think that the need for it has gone away. I don't think Juan Carlos is trying to say that, although it might have implied a little bit, but the models are good, but they lack some of the details. And that's because AlphaFold knows nothing about physics and chemistry. So anything that relies on amazingly, yes, but it's a tiny little Achilles heel of the whole process. Yeah. But they really accelerate what we're doing. So the models are almost always good enough that you can solve a crystal structure. And in fact, the young people at a workshop I was at recently introduced me to a new term, which is called alpha phasing. So that's when you
very good. Yeah.
phase the structure with AlphaFold.
Yeah.
And the only other thing I'd like to say is just to emphasize, as other people have, how useful and important it is that there are these good error estimates that allow you to say, let's downweight the parts we don't trust, let's break apart the pieces where we don't know the right interactions between them. And that's a really valuable part of the whole thing. Yeah.
Very good, very good. So another point that arose was about cross-linking. And Jan wanted to come back and just have a word about that to clarify his perspective on what he thought of cross-linking.
Exactly, because I don't want that anyone takes me wrong, and Yuri, I'm a big fan of crosslinks, and we use crosslinks, and we want to use more of crosslinks. So what I actually meant is exactly the opposite. So what I really meant is that now sometimes it seems that you can get away with using just one single data set. and doesn't have to be cryo-EM. It can be actually crosslinks without cryo-EM. So I think what you also observe, and what we also see, that sometimes you get enough of the answer about the structure just using AlphaFold and crosslinks together. You don't need now to spend all the time on the cryo-EM and optimization, which if you're interested in the general architecture and maybe sacrifice some details of the resolution that maybe you would get from crystallography or cryo-EM. So I think, yeah, what I meant for the integrated structural biology, it makes our work easier because we don't need to now really put all those data sets together in the program during the modeling process. We can actually use maybe 1 or 2 data sets for modeling and then the other data sets for validation, for instance. So yeah, still a big fan of crosslinks and want to use it more and maybe sometimes only.
Yeah, one additional piece of data we'll probably all need, and that is the stoichiometry.
Yeah.
You know, that cross-linking doesn't give you AlphaFold, doesn't give you EM, doesn't really give you, so.
Yeah.
And I can also certify from our test that sometimes, even if you try with AlphaFold different oligomeric states, you often cannot distinguish from the scores whether this is a pentamer or hexamer.
Okay, so
that would be a good something really important from experimental work.
So a related question came in saying sometimes repeat number is essential to fold. That's essentially knowing how many repeats. Does AlphaFold get this right more often than wrong? Or is that still a problem? Has anybody looked at that explicitly?
I looked at this a little bit when we were using domain motif resolved structures. Many of them, bind to a domain that is a repeat domain, like WT40 anchoring repeat domains. AlphaFold gets them right. Yeah, but we do see sometimes these domains often have multiple different binding sites. AlphaFold has sometimes trouble finding then the right binding site, but the repeat domain is really accurately predicted.
It's still accurately predicted. That's good.
Yeah.
Yeah.
Nuclear pore complex is full of repeat motifs, the domains with TPR repeats, WT40, and they are just fine. But indeed, sometimes when we have competitive interfaces, it can get confused.
Okay, so a general question that comes to mind really relates to the flexibility, a little bit what Katja talked about, but she sort of homed in on the small linear motifs, and some disordered regions just don't have those small linear motifs. So does anybody have any ideas about how AlphaFold might help in our explorations of disordered proteins? Hard question, I know, but flexibility. I was always taught whenever that flexibility was really critical for every single protein function, and to some extent that's true, but not actually. I think the reverse has been amazingly true from what we've learned about structure, but nevertheless, flexibility is important. Any thoughts on that?
I mean, all I know so far, and that's also from talking to other people who are using AlphaFold, who have been working in a disorder field, like doing predictors for disorder, they just say AlphaFold is better than existing tools in telling you here is a disordered region. But anything else beyond that, what AlphaFold would tell you is not reliable, essentially. Yeah, because it's trained right on structures, on folds. Yeah. It just knows that this thing doesn't fold. But the dynamics, there's nothing to take home at the moment from AlphaFold. That's my understanding.
I think I would agree with that. Any comments from anybody else on that?
Yeah, so one other thing that Katja said already is that in the context of an interaction, unstructured region can become structured. And it wasn't the example that I showed, but we also have an example where when we did the trimeric complex calculation, then the third protein actually winds around the other two, so on the surface along, and it's supported by our crosslinks. So that would be a fitting example for this question. Of course, that isn't dynamics, but at least it is how non-structured regions can become structured in the context of an interaction.
Yeah.
And the dynamic aspect, I mean, this is something that crystallography has been battling its heads with. EM, is moving up to seeing multiple conformers. Cross-linking is capturing it, but also suffering from it because it gets the superposition of all the conformations in the data, and then one needs essentially modeling to disentangle the overlaps. So I think we probably have a lot of young people in the audience. There is still enough space for research. Nobody needs to be worried that structural biology is going to be over in a few days.
There was one question that I suspect you answered about for cross-linking, how you differentiate inter and intramolecular interactions.
Yeah, if it's the same protein sequence involved, in fact, we often can't tell the difference. That's why we don't call them intramolecular, but we call them self-links to like stipulate. It could be homo-multimeric links, or it could be links inside a protein.
Yeah.
And modeling helps a great deal in sorting those, because if you have a model, then, well, if it doesn't fit within one protein, well, try putting a second there, and then you will see actually some of those really fit beautifully. Okay. And in this way, you do get out of the combination of cross-linking and modeling, not out of cross-linking alone, but out of the combination with modeling, you do get the distinction.
Okay, so could we just go round the table, and could people suggest the one thing they would like to see developed using deep learning, not necessarily for structures, but where, I think what's happened is that now everybody is thinking about deep learning almost as a shortcut to prediction for many of our biological problems. So if you could, have a magic fairy to imagine a new predictive tool using this deep learning, what would be the problem you would set? Yeah,
I can start because that's easy for me, I think, and this is already happening. So it's not like this super original idea, and we are observing this for some time, but we're looking forward for these methods to be really good. And this is about finding structures or molecules in cryo-electron tomograms in cells. So there's now a technology that can give appear with electron microscopes into cells, and we see ribosomes, proteasomes, nuclear complexes, and not much more, but those complexes are there, and most likely the signal is also there. And it seems that there's a consensus in the field that with deep learning, you can actually sieve them out. And even yesterday, there was a paper on bioRxiv published on that. So I was really looking forward to a breakthrough there.
So Jan, is that using image processing sort of approaches rather than sequence-based approaches?
Yeah, that would be more image processing, yes. Unsupervised detection of patterns, classification.
Yeah, I think that's really exciting. That might give us our jump from molecular to cellular seriously in a way. That would be fantastic. So I'm going to go along my list. Yuri, what would be yours?
Well, it would also be along the line of going from molecular to cellular, and that is cross-linking can be done in cells and has been done in cells. But the data we get is while we have ways of interpreting it, these ways are not, are still improvable in so many different aspects. There's so much more information in this data than what we are using currently. And it's not, as Jan already said, it's not like a far-fetched idea. And in fact, we are working on this as well, as probably others, to pull all these information sources that we do actually have on the data to get deeper into it and get a more complete description of the interactions and the structures of proteins in cells, which would beautifully team up with Jan's.
Yes. Randy?
Well, would it be nice to have a warning of this, but one that comes to mind is
No.
one that comes to mind is actually, being able to take deep learning models and learn something about metabolic interactions within a cell. So what are all of the networks and how do they interrelate? And can you learn to predict what happens if you inhibit one protein in the rest of the cell?
Yeah. Okay. Thank you. I don't. Is Juan Carlos still online? He did not.
Yeah.
Yes.
I'm here.
Sorry, you're off the end of my pictures.
Screen.
Yeah.
Yeah, well, I think that there are 1,000 challenges in pharma when we go from target identification until clinical trials. And nowadays, AI, machine learning have been, people is trying to apply them, and every single step of this pathway, let's say.
Right.
What is lacking is well-curated data. I mean, especially for deep learning, we need large amount of data, and I think that's the basis, of all those methods. I mean, we need the data first, right, and then think of the architectures, right, on top of it.
Yeah.
So I think that we are being lucky that the community of crystallographers have been curating data for 40 years, right? I mean, collecting in the protein structure database, high quality data. I think that will be one of the reasons, right, those methods have been so successful, right? Because the ground, the basis, right, is there, right?
Absolutely. Thank you, PDB, is all I can say.
Exactly. the bank, I mean, it's been very helpful to have well curated data there. So I think that we should write them everywhere, because even though we might accelerate, let's say, one step on these multiple challenges, it will remain other bottlenecks down the road. So we need somehow to, try to accelerate those, especially the slow ones. So, but I think it's going that direction. People is applying everywhere machine learning AI, trying to make every step faster, and that's what's needed, to really accelerate things. We need to improve the whole process, because otherwise we jump to the next bottleneck, and then
Exactly. Katja, how about you? Do you have a wish list?
Yes, I have two big wishes, and the first one is maybe one that's also I heard already being worked on by DeepMind. Is the prediction of the effect of mutations on protein structure and function. I mean, that would have such a tremendous impact also on translationally, right? On medicine, that would be amazing. And the second sort of maybe more far-fetched wish, which is in line with Randy's, is to get a predictive model that takes from the genotype to the phenotype. Right, we have a lot of data by now collected, right, kinds of omics data, but we struggle to integrate this and to make predictions, and probably only machine learning can solve this.
I think in that context, the fact that, in a sense, AlphaFold doesn't know any physics and chemistry, and yet it still can sort of jump over that to get that I never thought would be possible. I thought we would really need to know how the protein folds, etc. And the fact that it doesn't need that gives some hope that maybe some of these, like from genotype to phenotype, where there's so many steps in between, but maybe we can ignore them all and just jump straight over the top. But I think that perhaps is, well, we'll see, won't we? I think it's very exciting. Perhaps I could have my own wish list to add to this. Mine would be very much, given a protein, predict what its ligand is, especially the small molecule ligand. We haven't really gone into, I mean, obviously AlphaFold is based on the PDB and all the data that's captured in the PDB. And we know that the chemical space is so much bigger than the protein space. And yet that interaction with small molecules really is at the basis of life, probably more than protein-protein interactions, I think. And so if we could find those, then I think then it comes back to the metabolism and that side of things. I think it would be fantastic, but it's not clear to me whether this will work as well, partly because we don't have as much data, but also because the data space is larger than the protein space, which always really surprises me even now, given how big proteins are, and yet they're all kind of the same. They're all variations on a theme, whereas the molecules are much more challenging, the small molecules in a funny way to go. So one question I was going to ask Jan, actually, you've already mentioned, obviously you've worked on nuclear pore, Jan, but what are the other prime large complexes, and how the nuclear pore has a solid scaffold in the middle? It isn't a transient interaction per se. It has this solid scaffold. So which of the other potential complexes, where many of them are transient and passing, do you think this integrative modeling, using many of the things that we've heard about, will really pay dividends in the next few years?
Which exactly, or what types?
Well, no, I was thinking of, the big, I mean, you mentioned the proteasome.
I mean, definitely AlphaFold, integrative approaches with AlphaFold work better for more stable things, and also from some specific species like nuclear pore complex from human works much better than from some other eukaryotes. So that it's a really, difference, which difficult question, which complexes can be addressed. I dream about going bigger. So maybe growing the nuclear pore complex and adding all the other Turek complex, lamine, all these kind of things, and see where you can stop. With our lab, we work on other membrane coats, some other species, vesicles, influenza viruses as well. So these are
actually, there is a question about viral proteins. So I'll ask the question explicitly. I would like to hear from Dr. Rapp-Silber what he thinks about using AlphaFold to predict structures of proteins without significant alignments in any database. For example, a completely new viral protein without alignments. So, Yuri, do you have any comments on that?
Well, you can switch off multiple sequence alignments in AlphaFold, I believe. Yeah. And then the predictions become less good. Of course, you could imagine adding experimental data, which isn't possible right now, but crosslinks are very similar to the evolutionary constraint that you do get out of multiple sequence alignments. So maybe one could replace then lacking multiple sequence alignments by some experimental evidence. Yeah. And I seem to recall that in the first talk of the seminar series, somebody mentioned that there is something new coming out that isn't even on bioRxiv yet, that works without multiple sequence alignments. But Jan.
Anybody? Yes, I've heard that sort of rumor as well. Jan.
Yeah, I just want to comment from our experience with viral proteins or proteins that don't have many homologs. It seems that it depends on your luck, whether your particular sequence or structure follows patterns that are in PDB. So I think that might be that why human sequences also work better, because maybe there are more human and related sequences in the database. So it's simply how typical your protein is. And for smaller things, I think how much it follows usual statistical physics patterns, and we see it really depends on the case simply.
Yeah. Okay, so I think we're at 3 o'clock, so we should finish up. I'd really like to thank our panel. They've done a great job of presenting us. I think they also did a great job about dreaming about the future. And I would encourage everyone on the call to absorb what you've heard and to dream about how these methods might really be applied to many different aspects of biology. I think this is a real opportunity for us all to not just use the AI, but to incorporate it with all the other experimental data. We don't want to shut our eyes and just try to predict blank. We want to use everything that we've got. And so I encourage hopefully a lot of younger people on the call to go away and dream.